Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to class 9th and welcome to App6 online support program. Here's your biology teacher and I am Faranju. This is week 2, day 3, our lecture number 6 and our chapter number 6, that is enzyme. Let's recap our previous lecture, that is mechanism of enzyme substrate complex. We know that every block has a specific key. In the same way, every enzyme has a specific substrate for its binding. We have learned that first enzyme and substrate gets bind. After that, ES, enzyme substrate complex, is formed. After that, enzyme product, EP complex, is formed. Finally, the product is released and enzyme is used over and over again. Now in the next slide, we are going to discuss the two interactions or two models for enzyme action. That is lock and key model. In figure A, you can see a lock and key model, and the second one is induced fit model. Let's move toward our lecture. So today's topic is models of enzyme, that is lock and key model and induced fit model. Number first model is lock and key model. We discussed mention points. It is discovered by Ingier, definition and explanation. The first one point is discovery. It is discovered by Emil Fischer in year 1894. This is important for your MCQs. The next is the three terms. Dear student, three terms, that is enzyme, it is a globular structure, active site is present, it is a three-dimensional cleft, okay, of enzyme, and the substrate. Three things. So according to this model, active site of an enzyme is rigid. This three-dimensional cleft is rigid. It means a specific substrate get bind with a specific enzyme and it is converted into product. Only one substrate can transform into product. So students, our next topic is induced fit model. We are going to discuss the discovery, year, definition and explanation. It is discovered by Daniel Koshland in year 1958. Important point, highlight it, important for your MCQs. So the definition is, or according to this model, active site of an enzyme is not rigid like a lock and key model. Here you can see in figure number 1 that enzyme active site has different shape and substrate has different shape. You can see. In figure B, you can see that enzyme active site change its shape according to the shape of substrate, because active site of enzyme is not rigid. What forms? Enzyme substrate complex is formed. After that, third diagram shows or depicts EP, enzyme product complex. Finally, the products get released. Enzymes, again enzyme active site change its shape as in the figure 1 and it is used again and again. Okay. So this is induced fit model. Let's review today's lecture, that is lock and key model and induced fit model. According to lock and key model, enzyme active site is rigid. A specific substrate gets bind with the active site of enzyme, then ES complex form, then product is finally released. And induced fit model, enzyme active site is not rigid. It changes its shape according to the shape of the substrate, then enzyme substrate complex form. After that, product is released and enzyme again changes its active site shape. Okay. I hope all of you have understood the concept of both these two models, that is lock and key model and induced fit model. This is the working mechanism of enzymes. Now what you have to do, learn the lecture and read the topic from book and also highlight the important points mentioned in the book. You have to remember the discovery, dates and the definition of these two models. Okay, fine. If you have any query, then ask from me. Thank you, students. Take care and have a nice day.